So today we'll be diving into the molecular basis of cervical cancer. In our previous lectures, we'll look at what cervical cancers are, and today it is imperative that we'll see what happens at the level of the cell when it comes to cervical cancers. So the causative agent for cervical cancer is persistent infection with high risk human papilloma virus, which is the key factor. Type 16 and 18 cause about 70% of cases. Human papilloma virus infects the squamocolumnar junction of the cervix. So the oncogenic mechanism of human papilloma virus is essential, and this is what happens. Human papilloma virus produces oncoproteins E6 and E7 that disrupt normal cell cycle control. So the E6 protein binds and inactivates p53 tumor suppressor. This prevents apoptosis and DNA repair. This results into cells with DNA damage that survive and proliferate. So the E6 protein binds to p53 suppressor gene and then prevents apoptosis and DNA repair. So cells that are damaged and have bad DNA will continue to proliferate. So the E7 protein also binds and inactivates retinoblastoma protein, which releases E2F transcription factor. This results into uncontrolled progression into S phase, resulting into increased cell proliferation, which is the hallmark of malignancy. So in a nutshell, E7 binds to retinoblastoma protein, releases E2F transcription factors, resulting into uncontrolled progression into S phase, which will further result into increased proliferation of cells. So the activity of the E6 protein and the E7 protein result into genomic instability and uncontrollable cell division. Additional molecular changes also occur, such as activation of telomerase, which causes immortalization of cells. Epigenetic changes, such as promoter hypermethylation, which silences tumor suppressor genes. Mutations in host oncogenes and tumor suppressors accumulate over time. Angiogenesis stimulated via VEGF upregulation. So here new blood vessels are formed and blood supply to the new highly proliferating cells. So the disease continues with its progression. Persistent human papilloma virus infection will definitely result into cervical intraepithelial neoplasia, which is known as CIN. So CIN1, which is mild dysplasia, that's low grade and may regress. CIN2 to 3, which is moderate to severe dysplasia, and it is referred to as high grade. Carcinoma in situ, which results into invasive cervical cancer, which is squamous cell carcinoma, most common, or adenoma carcinoma, which is less common. So what are the key risk factors for persistence/progression? First is early onset of sexual activity, multiple sexual partners, immunosuppression, e.g. in HIV infection, smoking, which causes synergistic DNA damage, long-term oral contraceptive use, which causes weak association. In summary, cervical cancers develop due to persistent infection with high-risk human papilloma virus, whose E6 and E7 oncoproteins inactivate p53 and retinoblastoma gene, leading to genomic instability, dysplasia, that is cervical intraepithelial neoplasia, and progression to invasive carcinoma. Thanks for listening.